Hello, everyone. My name is Varanya. I'm the Senior Scientific Coordinator of the GenRe-Mekong project. Just a quick intro about the video that you will watch very shortly. It is a recording from the Genetic Epidemiology Journal Club organized by MalariaGEN. Do check out the MalariaGEN YouTube channel for more of the recordings from this journal club. If you want to go straight into the talk, you can use this timestamp here to jump straight into the beginning of the talk. The talk that you will listen to very shortly will explain to you a little bit about the GenRe-Mekong project and why we started the outbreak investigation and why this use case is important and relevant to the regions and countries that are approaching elimination, and how we can use the genetic epidemiology to characterize outbreaks. It will then share the findings that we have published on this investigation in the Lancet Infectious Diseases in 2022. Thanks for listening and enjoy.
So first of all, I wanted to say, that I'm very happy and honored that we are starting this new series of journal clubs. The idea is to bring partners together, is to bring people that are publishing in malaria field. It can be parasites or also can be vector, of course, human. And to have a space here for us to discuss the paper, discuss their findings. And we have more of a collective journal club than something that happens isolated in people's own labs. Obviously, these are always very helpful as well, but we thought that we have such a large network, we collaborate with so many people, that would be an excellent opportunity to bring everybody together and share our findings, share discussions, and just share. So given the relevance and the excellent work that Varanya and Oliver did for the Laos paper that was published earlier this year, we thought that would be an excellent case to start with Varanya presenting on the work. Varanya is working with Oliver in Bangkok in Moro, and she's a senior scientific coordinator. I met Varanya a few times, and and her work is just amazing. She's done an excellent workshop with NMCPs as well, showing them how to interpret some of the genetic data that Oliver produces. So it's just all excellent work from Varanya. So Varanya, thank you so much for your time, for your effort, and for your willingness to come and be here as the first person to give us the journal club. And also, I wanted to say that if other people, ideally we would have this on a monthly basis. So if people wish to present something, just let me know, and we are more than happy to include you in the next round as well. So Varanya, off to you, please.
Thank you so much for your invitation. I am very excited to share with you about our work that we did on the outbreak characterization. Let me share the slides with you and move the video away. It's blocking me. Okay, can everyone see my slides? I hope so. Okay, so today I will be sharing with you some of the findings that we did on the outbreak characterization last year. But before we move on to talk about the findings, I am aware that perhaps the audience may be a bit different, people from different groups. So I wanted to start this talk by introducing to you about who we are, the GenRe-Mekong project. And then I want to talk about why we we started the work on the outbreak characterization use case, and why this use case is an important use case and very relevant to regions that are approaching elimination. Then I'll explain how we can apply genetic epidemiology to characterize outbreaks. And then we'll get into the findings, the results from the Attapeu outbreak, which we have published this paper last year. So the GenRe-Mekong project is a malaria genetic surveillance project operating in the Greater Mekong subregion. It is a partnership project that brings together scientific capacity and partnering with the control programs in the endemic regions. The project is led by Professor Olivo Mioto, and we are based at the Mahidol Oxford Tropical Research Unit in Bangkok. So the purpose of this project is to provide timely and useful information about the malaria parasites in the endemic region to support national malaria control programs in their decision-making regarding elimination of malaria. And in order to provide comprehensive information, we need to collect a lot of samples. And the best way to collect, to do this is to partner up with the control programs and collect samples at health facilities. Well, this map shows the collection sites. It is rather an outdated diagram, but it gives an idea of the coverage of the collection site that we have. In the earlier phase of the project, we worked with research projects, but since 2017, we have been working with the control programs to cover routine genetic surveillance, and we have been actively operating in Cambodia, Vietnam, Laos, and starting this year, we will be also starting the Thailand project. The project is funded by the Bill and Melinda Gates Foundation and the Global Fund. Once we have collected the samples, we collected the samples in a form of dried blood spots. These samples get sent to the labs, and we then get the information about the parasites. The information that we get are information such as whether the parasites are resistant to the drugs, whether they are related to each other, whether they are spreading, for instance. GenRe-Mekong then analyzed this data and communicate this information back to the control programs, hopefully to support them in their decision making. And so at this point, you might be wondering what's the malaria cases like in the Greater Mekong subregion. So the Greater Mekong subregion has seen a big reduction of malaria cases over the past few years, which is a great thing for the region because we are on the right track. But in a few isolated pockets of Laos, we have seen an outbreak happening. And we know that there has been an outbreak because in a few part of the country, there has been a sharp increase in numbers of cases, while the rest of the countries, the numbers are low. So as countries and regions are approaching elimination, we would see more and more outbreaks happening. Outbreak can cause resurgence of malaria and can hamper progress of elimination. A malaria outbreak can be defined as a sudden and rapid increase in the number of infections, often localized at a particular site or district. A criteria that control programs may use to assess whether there's an outbreak could be that they might be comparing the number of cases in a particular area compared to the average of 3 to 5 years. However, there are no globally accepted criteria for declaring an outbreak, and different authorities may be using different criteria. And so the criteria for declaring an outbreak is all up to the control programs. Once the control programs detected that there has been an outbreak, they might be wondering, why did the outbreak occur? Was it because of heavy rain, or more people going to the forest, or was there more mosquitoes, or was it because of resistant parasites, or whether the drug is failing or at risk of failing? Being able to distinguish what is driving the outbreak can help control programs to have the most effective response. And so the outbreak that we characterized was in a province in the southern part of Laos, in Attapeu. And Laos is a country in the Greater Mekong subregion, and it is aiming to eliminate Plasmodium falciparum by the end of 2023. And since 2004, Artemether lumefantrine has been the line treatment for falciparum. The malaria is endemic only in 5 provinces in this country, and they're all in the southern part of the country. And the cases of malaria has been declining. But in the year 2020 and 2021, the control programs has noticed a sharp rise in the number of cases in Attapeu. And so they have asked us to investigate into this outbreak. So Attapeu has seen sharp rise in the number of cases of 130% increase, while the other endemic provinces have seen an average of a reduction of 65%. Attapeu is a largely forested province with low population density, and it is bordering with Vietnam and Cambodia, and there's little international traffic into this province. Malaria is mostly transmitted in the forest, and it is mainly affecting forest workers and their communities. So what facilitates this outbreak investigation is the collaboration between partners. It all started with SEMPE, the control programs of Laos, noticed the unusual increase and declared that there has been an outbreak. And they have asked us to investigate into this outbreak. Then the samples are then collected by ROMRU and sequenced by the Sanger Institute and analyzed by the GenRe-Mekong project. The findings are then communicated back to the control program to support their responses. But in this talk, I will focus on this part of the flow of events where we characterize the outbreak. In the GMS countries, a typical assessment of outbreak normally uses clinical or patient travel data that are reported by the health facilities. This can be challenging in many ways because it is quite common to get report biases, and also patients are not always willing to tell about their travel history for many reasons. And so in our work, we use a different approach. We use the genetic epidemiology to support assessment of outbreak. Genetic epidemiology often focuses on the genetic factors and then apply that to the control of disease. And in this part of the world, malaria outbreaks are often ascribed to the increase in transmission due to factors that are related to ecology, such as increase in mosquito activities or the weather condition, or to or ascribed to the human behavior, such as increased traveling to the forests or importation. But outbreak may also be caused by the rapid expansion of parasite population driven by selection for genetic traits that increases its chances to survive or transmit. Being able to distinguish the different type of outbreaks then is quite important. And so how can we use the genetic information to help us to determine what is the main cause of an outbreak? So outbreaks that are driven by selection versus outbreaks that are driven by increased transmission, such as increase in mosquito activities, would have different genetic characteristic. And in the next few slides, I will try to explain this concept with some simplified diagrams. In normal circumstances, when there is no selection, the malaria parasites are expected to sexually recombine in mosquitoes, which maintains the population diversity. When anti-malaria is being used, this applied selective pressure on the population, and eventually mutation that helps the parasite to be resistant to this anti-malaria will arise. And then this parasite will then be able to survive the anti-malaria in use and thrive and multiply while other parasites get killed off. This leads to reduction in the population diversity as many identical individuals expand in number. And so an outbreak that is driven by selection might be characterized by a significant reduction in population diversity compared to the period before the outbreak, decrease in heterozygosity, and expansion of large clonal clusters. As for the outbreaks that are driven by increase in vector activity, we would have expected a different genetic profile. So in moderate and or low transmission settings, such as in the greater Mekong subregion, a person is expected to be infected by only one type of parasite. A mosquito may decide to bite more than one person. When there has been an increase in vector activities, but when there is no selection or preference towards one type of parasites, we would expect to see an increase in case numbers, but the diversity would be expected to be equivalent to the period before the outbreak. In some situation, we might expect it to see an increase in population diversity because increased in vector activities would increase the chances of outcrossing, which forms recombinant parasites, such as the example here. And so outbreaks that are driven by increased vector activities or human movements would maintain or increase the outcrossing rate, heterozygosity, and allelic diversity. So having these concepts in mind, we applied this to our work on the outbreak characterization. In a lot of the things that GenRe-Mekong do, we often started by considering what the program use cases are, like the public health activities, and then we would see what genetic epidemiology methods can be used to support the program use cases. There is a great resource on this on the MalariaGEN website. The document outlines how genetic epidemiology can be used to support public health decision-making. But for this talk, I will focus on the response to outbreaks use case. For this use case, there are 4 methods that support this investigation. So just as I've shown in the earlier slides, we can assess the changes in diversity level to detect changes in population structure. We can detect whether an outbreak is caused by clonal population expansion. On the other hand, if we don't see any, if the diversity has been unchanged, then the outbreak could be caused by increased transmission. We can also monitor genetic markers to assess the spread of resistant alleles in the region and identify circulating strain by detailing the known phenotypic traits. We can also identify some imported cases. In the Spot Malaria platform, which is the platform that we based our genetic surveillance on, there are 2 key features that help with these analyses. So the first feature is that we genotyped many drug-resistant markers which support this analysis. The other feature is the genetic barcode, which help to support these 3 other analyses. So the table on the left-hand side shows the drug-resistant markers that we genotyped. For each of the samples, we also have the 101 SNP barcode, which has been genotyped across the falciparum genome. The barcode is essentially like a summary of the genome, and we can use this to do analyses such as assessing population diversity. We can see whether the parasites are related to one another. We can track the transmission routes, for instance. Okay, so now I'm going to start talking about the findings from this paper. In our investigation, we compared the outbreak samples, which was collected in Attapeu during the 2020 and 2021 season. And we compared that to the samples before the outbreak. We compared that to the sample before the outbreak, which we have been doing surveillance since 2017. So we can compare that to the other endemic provinces. We also have some samples from Attapeu from earlier, from 2011 and 2012. And so what we did was we compared the diversity levels and also identify whether there's any expanding group. But this list over here is pretty much similar to the list I've just shown you previously. We also monitor the genetic markers and predicted the characteristics. We also analyzed the relationship between groups, and 4 of these analyses were done on the amplicon sequencing surveillance data. And in order to dig deeper into the origins of the strain, we also used whole genome sequencing to track ancestry. So we started our investigation by comparing the diversity level. We used the barcode heterozygosity as a measure of diversity. So in Attapeu, we compared the diversity level across the years, and we can see that from 2011 to 2019, the diversity level is relatively stable. But starting from 2020, there has been a big reduction in the diversity level, such that in this period, it was significantly lower than what we have seen in Attapeu previously. We also compared the diversity level of Attapeu samples to the other endemic provinces. In the period before the outbreak, the diversity level in Attapeu was actually slightly higher than the other two provinces. But during an outbreak, there has been a massive loss of diversity, which is unprecedented for Attapeu and for Laos in general. We expect that this massive loss of diversity could be caused by an expansion of identical subpopulation. So we investigated into that. In order to group together identical parasites, we calculated the pairwise genetic distances, and we used this to cluster the parasites together with at least 95% barcode identity. We use 95% barcode similarity instead of 100% to account for any genotyping error. But the point is that we wanted to group together identical samples. This analysis identified 30 clusters, and we plot that on the proportional bar charts. In the period of 2011 to 2012, we could see high diversity level such that at least 88% of the parasites did not cluster with any other parasites. They have unique barcodes. And as we move into the 2017 and 2018, we started to see more population structure emerges, as shown by the division in the bars. And the unique barcode dropped to about 31%. During this period of in the Greater Mekong has experienced a there has been the KL1PLA1 has been prevalent in this region and has also entered into the Attapeu province. And starting in 2019, we started to see 3 groups expanding, such that, and the most rapidly expanding cluster is the LAA1. It expanded quite rapidly to such that 84% of the population in Attapeu belongs to this cluster by the end of 2021. So the LAA1 cluster, they carries the R539T Couch13 mutation, which is resistant to artemisinin. And a few of the samples in this cluster has the MDR1 amplification, which is a marker for mafequine resistance, as well as there has been some association with reduced sensitivity to lumefantrine. The other outbreak cluster, that LAA7, also carries the R539T Couch13 mutation. And both of these clusters were found only within Attapeu at the time of the analysis. The last outbreak cluster is the LAA2. This cluster carries the C580Y Couch13 mutation, and only a few samples has the resistant to piperacin, and so this cluster is not the KL1PLA1 strain. LAA2 has been can be found across the region, and it has been circulating at low frequency in each of these sites. So many of us are quite aware of the KL1PLA1, which carries the Couch13 C580Y mutation and also has the amplified plasmapsin 2,3 gene. So it is resistant to the DHA piperacin. During the period in which in this country have been using the DHA piperacin, KL1PLA1 was very prevalent, and it has entered into Laos even though Laos does not use DHA piperacin, and it has entered into Champasak and Attapeu province. And when we look at the population in Attapeu, indeed, we did see that during this period, 2017, 2018, many of these samples were the KEL1 PLA1 carrying the C5ATY and amplified plasmapsin 2-3, as shown in the red. As we entered into the 2019, we could see that the KEL1 PLA1 just disappears from the population. This coincides with the changes in drug policy in the neighboring countries of Laos. So Vietnam, Cambodia, and northeast of Thailand have changed from the use of DHA piperacin during that period. And this is the prevalence map of DHA piperacin resistance in the GMS. It is not in the paper, but I just wanted to show you because it's data from the GenRe-Mekong project. We can see a rapid decline in resistance to DHA piperacin in the region soon after the switch from this DHA piperacin. And this is also, of course, the decline in resistance is also a disappearance of the KEL1 PLA1 strain. And we can see that starting in 2020 in Laos, we barely see any resistance to DHA piperacin, and which means also there's not going to be a lot of KEL1 PLA1 as well. When we look back at our Attapeu population, and we could see that, yeah, the KEL1 PLA1 is not there, and the disappearance of KEL1 PLA1 has left a vacuum in which this strain LAA1 could expand. And so we can see a shift in KELCH13 dominance in Attapeu from C5ATY to the R5390. Then after this analysis, we wanted to understand and see whether what is the relationship between the different strains are. And so we did this by plotting the genetic distances between a pair of individuals in Attapeu using the genetic barcodes. Well, heat map often has a lot of information, so I just want to kind of introduce you a little bit on how you can read the heat map. So in this heat map, if you see more of a red color, it means that the pair of samples are more distant from one another. If you see more of the blue color, it means that the pair of samples are more similar. At the level of distance of 0, it means that all of the SNP barcodes are matches between that pair of samples. And at the top of the heat map, I have put in some information about the samples. We have the year of collection, the KELCH13 genotype, plasmapsin 2-3 amplification status, and which cluster they belong to. So what is quite striking when you first look at this heat map is, of course, you could see that this LAA1 is very highly clonal and inbred. There are very little evidence of recombination with other samples from Attapeu. This characteristic of the LAA1 can be contrasted with that of the KEL1 PLA1, which forms a fragmented group of fragmented clusters of different sublineages. What is interesting is that the LAA7, which has the R5390 KELCH13 mutation, is also located within this KEL1 PLA1 cluster, and the genetic barcode shows that it is highly similar to the K1PLA1, in which around 75%, 80% of the barcode matches the K1PLA1. On the other hand, the LAA2, which has the C580Y but without the amplified plasmapsin 2,3, it is not that similar to the K1PLA1, so it is distinct from this group. So seeing this relationship on this heat map has raises a few questions for us regarding the origin of the strains, whether the LAA1 has emerged independently or whether it has derived from an earlier R5390 lineage, and whether the recombination event could explain this relationship that we see between the LAA7 and the K1PLA1. And in order to do this, we needed to use the whole genome sequencing. So from the whole genome sequencing data, we carried out the identity by descent analyses, which identify the genome stretches in a sample pair that are statistically likely to have inherited from a common ancestor. The whole genome sequencing data was from Laos and also neighboring countries, and has been that was contributed to the MalariaGEN community project version 6.4. Here's a summary of the origin. So when we first started this analysis, we tried to find the most earliest samples that we have that carries the R5390 and C580Y. And we have found two founder population, which was previously given a name KH3 and KH2. So what we found was that LAA1, it inherited a large IBD region from the KH3, and there was the large IBD stretches and the limited linkage breakdown indicates that there has been very few recombination events has occurred in the LAA1. For the LAA2, genome is pretty much identical to the founder population KH2B. And so this lineage has been circulating in the region virtually unchanged for the last 10 years. And this lineage is related to the K1PLA1, but it is distinct from it. And then for the LAA7, it is a recent recombination of two local strains. One of them is a K1PLA1, which also derived from the KH2A. And a large portion of the genome comes from the K1PLA1 lineage. And the part of the genome where KH13 gene lies comes from this KH3 lineage. And so what this confirms is that the IBD analysis from whole genome sequencing really do support the relationship that we observe in the genetic distance heat map that was plotted using the SNP barcodes. So in conclusion, all genetic evidence is pointing to a selective sweep of LAA1 being the primary cause of the outbreak. The significant reduction in diversity in the population and the rapid clonal expansion is characteristic of population that undergoing strong selection. If this outbreak was driven by other factors such as increased vector activities or increased traveling to the forest, we would have expected more recombination in the data and more and higher diversity, but that is not what we observed. And even though there are a few other varieties of parasites in the region in the same time, LAA1 did not recombine with those other genetic background. It could be because so the low level of recombination in LAA1 suggests that that mating with genetically identical background would help to retain the selective advantage of LAA1, while mixing with other genetic background would have disrupted this advantage. But sadly, our data cannot pinpoint the exact nature of the selective advantage. The selective advantage could be anything that helped the parasites to survive and transmit. But given the history in the Greater Mekong subregion, it is probable that drug resistance could be the cause. Hypothetically, LAA1 could have reduced sensitivity to lumefantrine, which is the ACT partner drug that is being used in Laos. But our data cannot confirm this hypothesis because we do not have globally accepted a marker for lumefantrine resistance. And to go back to this flow of events, once we have determined the cause of an outbreak, we communicated this finding back to the control program and other public health stakeholders relatively quickly before the next malaria season. And as a response to the outbreak, SIMPE, the control program of Laos, have carried out active case detection, and they have started the therapeutic efficacy study of artemether lumefantrine in Attapeu, Sekom, and Sawanakhet. And before I finish, I want to leave you with 3 take-home messages. So first, the implication of the data. So the implication of the data shows that it is crucial for us to monitor all the KH13 mutants rather than focusing on variants whose dominance might be temporary. In our data, we have shown that malaria lineages can be very resilient, and they can be just circulating in very low number for a long period of time before they take over when the condition is right for them to outcompete other strains. Second, the power of genetic surveillance. I hope that this work has demonstrated to you that genetic surveillance, which is relatively straightforward and affordable to implement, can address important public health use cases that are beyond monitoring the drug-resistant markers. And here, this work has shown that it is a powerful tool to characterize and analyze outbreaks. And then third, I hope that this work has shown you that the strong partnership between researchers and public health can provide highly informative data and timely data to support public health use cases. And so we encourage the research community to continue to share data to support these and other important public health use cases. And lastly, without a great network of collaborators, we would not be able to do any of this. So I would like to thank you, the Bill and Melinda Gates Foundation, the Global Fund, and the Wellcome Trust. I am not able to list out individual person's name on the screen, but I just would like to thank you all of our collaborators in the different research institutes and the control programs. And yeah, thank you so much for listening.
Thank you so much, Varanya, for the excellent talk. It was very inspiring to hear, particularly this very last remark and all the results that you had. Very clear, very constructive, just excellent. Thank you so much. So we have 14 minutes for questions. If anybody has any comments. Actually, Oliver, do you have anything to add?
No, I think Varanya has said absolutely everything very, very clearly. But we should see whether somebody has comments from the audience.
Yes, please. Yeah.
Other people.
So we have the first. Oh, sorry, it's not. Will Hamilton wants to add something.
First, just say that was an amazing talk, and it's a great paper. I think it's one of my favorite papers ever. It was just really, really good. I was going to ask, so the WHO definition of surveillance is to monitor the effect of a control program and also to try and detect early warning signs of threats to control. And in this case, it seems like the action was all initiated by the recognition that an outbreak was already happening. And I wonder have you thought about or tried retrospectively seeing, could you have predicted that an outbreak was happening before it was so obvious to the control program, whether that's by complexity of infection or diversity or something in like 2019? Could you have seen it coming, or how could you try and work on that kind of predictive power? Thank you.
Thank you so much for your question, Will. That's really, good question. I have to say that we have not looked into how the prediction of the outbreak, but it is a really good thing to look into.
Can I just add that we do have, we are aware of a number of other outbreaks that we have data for. And one of the things that we would like to do is to try to understand the difference between them. And maybe that might give some clues into how to predict them. And Jacob here has said that he will use machine learning to predict them. So we are on a good thing.
It's recorded, Jacob.
Thank you.
Thank you. So there's a question on the chat, Varanya. So is that have you seen or planned for diagnostics resistance for HRP2 and 3 deletion selection study in addition to drug resistance?
Since our project is very much focused at the moment on the greater Mekong subregion, we do not really see this problem with the HRP2/3 deletion. So we really have not looked into that yet. Sorry.
Thank you, Varanya. So I'll go to one question from the chat, then one from the audience. Alfredo.
Yeah, Varanya, congratulations for the presentation, the work, all the team. I think you are inspiring many of us working on this field. So yeah, very happy to see how the work is going. My question is more practical. So my experience here in Mozambique, where I'm working, is that it takes still a little bit of time since we collect the samples until we can provide a response to the program. And do you have any recommendation or how to improve to make this faster? I understand that it took like 1 year for you since you collected the samples to you produce the results and communicate it to the program. Any tip, any recommendation on how this could be even faster to make a higher impact? Thank you.
Thank you. I think maybe the best person to answer that would be Olivo. Do you have any tips?
Oh, thanks for that. Yeah, Alfredo, you are embodying my own concerns here. I mean, it's difficult, and it very much depends on the country you're working in, and it very much depends on on how efficient the sample handling and collection network is. I mean, I believe Mozambique probably has quite challenging geography, and so I think in our case, what was very helpful was in fact that this was initiated by the control programs, and so they supported some sort of emergency collection activity, which did shorten the time to get the samples to the lab. Also, you know, Sonia's team here at Sanger did give priority to the samples. So it was a bit of an ad hoc process, and I'm not entirely sure at this point how you would generalize it and make it more operationally repeatable, as it were.
Thank you. Thank you, Alfredo. Rania, another one. Yana, one, you've been, sorry, I think you had your hand up and I missed, you put your hand up before Alfredo.
Hi, that's okay. I just have two quick questions though. Like, I have no experience in genomic surveillance or GWAS study. So I like to know, like, how long would it take for like a new population to settle in the area? Because I see like you have the baseline from like 2012, but then your new sample from like 8 years later. So,
So your question is that how long does it take for a population to establish itself? Am I understanding correctly?
yeah.
I would say that from our data, the malaria parasites they adapt very quickly, and so as you have seen in the slides where I've shown, when there's a switch in the policy in the frontline treatment, you also see that it's affected the parasite population quite quickly. Yeah.
Thank you. And I have like another question regarding the emergence of the LAA7. If I understand it correctly, so it's like so it switched from like C580Y to 539, right?
It didn't switch. It's
Okay.
a mixture. So a large proportion of the genome of LAA7 came from the C580Y, like the K1PLA1, but a small portion of the genome was inherited from the R539T KH13 mutant.
Because it's very interesting that like crossing over in the gene, like the KH13.
That part of the genome did not cross over. It's the KH13 gene has been inherited just from the R539T lineage, but the rest of the genome that is not the KH13, where the KH13 gene lies, is inherited from the K1PLA1.
I see. Thank you. Because that's interesting. Because like there was like a paper from FreeDoc group that say like the one like haplotype of the KH13 would be better support from like one type of genetic background. So it's interesting that there was like increased emergence of like new KH13 mutant in the new genetic background, if I understand it correctly. Yeah. Great, thank you.
Thank you. Thank you for your question.
Thank you. So as I said, I'm just going to go to the chat because I'll alternate. So there's one from Tabitha. And they're asking whether are we able to use the same concept to monitor vector behavior and resistance to insecticide?
I have to admit, I have no experience in vector behavior at all. But I think, so I'm not really sure how to answer the first question.
And the second question is, can this data be used for prediction of future drug resistance possibilities?
Can this data be used? I would say that when we continue to do genetic surveillance, there are some things that we can use to help us to see whether there might be some selection going on in the population. And one of them could be the diversity, the changes in the diversity level. And yeah, so that can really help.
Can I just add to this that one thing that we couldn't achieve in this particular case that I would have wanted to do was to actually collect some of these samples from these expanding populations in order to phenotype them in vitro, because it is entirely possible that they, for instance, they have reduced sensitivity to other drugs that are used in Laos. So there's probably added activities that you could have once you get a signal from genetic surveillance. There could be other activities that follow on from there. Again, it's the sort of thing that you kind of need to have planned beforehand. We're sort of learning from this process.
Thank you. And last question from Hazel, please.
Thank you very much. Thanks, Varanya and team, for the very exciting work. I'm Hazel, all the way from South Africa, so just next door to where Alfredo is. So just out of interest, I can imagine that the people in the malaria control programs are not all scientists. So I'm wondering, how did you package the data for them to make it palatable and easy for them to understand? Did you prepare a one-page report, or how did you do it?
Yeah, so we often would write a report, and we would use a lot of maps to show the changes in, let's say, for example, resistance. We try to remove a lot of the technical jargons as much as we can without losing the important information. So we try not to, we try to only communicate the information that is relevant to the control programs.
Okay, so did you have training with them before that for them to sort of know what to expect when they see the report, or I don't know?
Well, recently we did have a training, but in the past we try to really use more of the visualization that are intuitive to understand, so for example, like the maps that I've shown you with the changes in resistant level. So is there anything else you want to add, Oliver?
Well, I mean, in this case, and it normally is the case when we deliver these reports to the control programs, but in this case in particular, we actually had a couple of calls where we discussed in detail the results with the director of the control program and the staff there, because obviously there were a lot of concepts there that they would not be very familiar with, and not just the genetic mutation and all this, but of course, explaining why we thought the expansion meant that it wasn't just sort of higher transmission from more mosquitoes and so on. That was a challenge to convey. So we did it through meetings and discussions.
Okay, thank you very much.
Sorry, Ethan, we've just reached to the 12 o'clock mark, so we need to stop it here. But I just wanted to say, Varanya, once again, thank you so much for the excellent talk, the excellent work you have been doing. I would agree that you have definitely inspired us today, and actually you've been inspiring us for ever since you published that paper. So thank you so much, and then thank you so much for everybody who came over and for Pablo to organizing this. And I hope to see you in a month's time. And again, if you wish to present something, and we're more than happy to host you. It could be a vector, it could be a parasite meeting presentation. So thank you so much for everybody that they are here. Have a lovely rest of the day. Thank you.
Thank you so much.